So Katia, I see that you have your screen shared in full screen. We can see your pointer. Everything seems good. So
Great.
welcome, and the screen is yours.
Great, thanks. Can you hear me?
Yes, we can hear you.
Great, fantastic. All right, well, thank you everyone for sticking through today. The title of my talk has changed very slightly. So I'll be talking a little bit first about tree-based phylodynamic inference, and then also some tree-free phylodynamic inference methods that we've been developing over the last few months. And this is all for emerging viral pathogens. So yes, you will hear a little bit about SARS-CoV-2. Okay. Whoops. There we go. Okay, I just want to start off with mentioning collaborators on the projects that I'll talk about today. The phylodynamic inference analysis that I'll talk about briefly is in collaboration with Adi Stern's group at Tel Aviv University, and involves Daniel Miller, who's a graduate student in her lab, as well as Mike Martin, who's a graduate student in my group. And if you have any questions about this work, it's been published a couple of months ago. The second thing I'll transition into, the second project I'll transition into is on tree-free phylodynamic methods development. That's coming out of my group with Youngsung Park spearheading the effort, and also Mike Martin being involved in that as well. So Samuel's talk this morning was, I thought really interesting. Well, this morning for me, this afternoon for you guys. And so he's really introduced some of the really kind of seminal approaches that have already kind of been used for phylodynamic inference for viral pathogens. So I'm going to just briefly review those here in a couple of slides. So as many of you know, sequences, viral sequences, or sequences more generally, can be used to infer the dynamics of spread within a region. And so this is an example actually from Samuel's group, where they've used some of the birth-death phylodynamic inference methods that have been developed by Tanya Stotler and colleagues on data from France. And what they did here for SARS-CoV-2 is that they estimated the R0, or the basic reproduction number, during certain time periods in France. So you can see here, higher R0 before the lockdown or the quarantine, and followed by a lower R0. Following that, and those estimates were based on viral sequence data from France. So that's using a birth-death phylodynamic model. The second approach, as Samuel also mentioned this morning, really comes from Eric Voltz's group. It's a coalescent-based approach, and so this is just an example using that coalescent-based approach. And I think these underlying really elegant mathematics that Eric Voltz has developed to actually estimate also kind of R0, or the effective reproductive number, kind of over time using sequence data. So both of these methods, the birth-death and the coalescent-based methods, basically rely on inferring a tree and inferring kind of the population dynamics, right? Underlying viral spread, and of course, the tree topology and the branch lengths and so forth are the branch lengths in particular are affected by the inference of the population dynamics as well. So we've been, over the last kind of few years, we've been really kind of interested in these kind of coalescent-based phylodynamic inference approaches. And so we actually use this really nice package that Eric Voltz has kind of published on a couple of years ago called phyldyne. I think this is the one that Samuel has not used. So we actually use this approach to study SARS-CoV-2 evolution within Israel. This is again with Adi Stern's group. So we looked at 212 full genome SARS-CoV-2 sequences, which were sampled between the end of February and the end of April 2020. And so you can see these sequences kind of here with these blue dots at the tips. One thing you'll notice is that there's many different independent introductions into the state of Israel, right? As was seen, in very many different locations. We saw it kind of in New York City, it was seen in Northern California, in Brazil, et cetera. It seems like the borders were very, permeable, I guess, pervious. So we saw many different introductions into Israel. And so what we were interested in as kind of our part of the analysis was trying to understand, or trying to estimate, R0 within Israel. And so again, we used Eric Voltz's approaches here. So we developed this underlying epidemiological model, which is an SEIR type model. So we have susceptible individuals, We have exposed individuals who will become infectious and infected, but are not yet infectious. And then we have these two classes of infected and infectious individuals, and then recovered individuals. Here we have these two classes, individuals who are infected and have high transmission rates, and individuals who are infected but have low transmission rates. And the reason why we incorporate these two classes is because we know for SARS-CoV-2 and many other viral pathogens, there can be a lot of transmission heterogeneity. And of course, with transmission heterogeneity, that effectively affects the variance in offspring distribution and patterns of coalescence. And so we had to kind of integrate the possibility for this transmission heterogeneity in the model. The way well, let me just kind of continue going on about the model. So we have a proportion pH here of exposed individuals going into this highly infectious class, and the remaining proportion transitioning to this low infectious class. And we parameterize the relative transmissibility of these so that the highly infectious class was responsible for 80% of the secondary infections, such that this pH parameter, let's say it's 0.2, that means that 20% of the most infectious individuals are responsible for 80% of the secondary infections, right? So that's that 20-80 rule that some disease ecologists use. If pH is 0.8, that basically means there's no transmission heterogeneity. And so the lower the value of pH, the greater the extent of transmission heterogeneity. So we were unable to kind of estimate this pH parameter, but all of our results kind of show parameter estimates, according to an assumption of pH. So I'll show that to you in the next slide. So the one other aspect that we have over here is this outside reservoir, because if we look at a phylogeny of the sequences from Israel, what you see kind of going forward in time, you see introductions into Israel from outside sources, right? So that these would be outside reservoirs, and going and so going backwards in time, we of course have to consider the possibility of these lineages in Israel actually kind of leaving Israel, going backwards in time. So that's why we have this outside reservoir that Eric Volz has also used in his analyses. And then finally, we allowed R0 to change on March 19th when lockdown or when the social distancing measures were put into place. Okay, so these are some of our results based on those 212 sequences. And so here again, we have this parameter pH with a pH of 2, meaning that 2% of the most infectious individuals are responsible for 80% of the secondary infections, and here all the way up to 80. So this 20 here is kind of that 20-80 rule in disease ecology. And so you can see across this range of transmission heterogeneity that we estimated R0 to be approximately between 2 and 3 initially, so before March 19th. And then after the lockdown, R0 decreased substantially and became about 30% of its initial value. Okay, so that's very consistent across the different levels of transmission heterogeneity. But we are interested also just in how much transmission heterogeneity there was actually. So we actually just considered reported incidence by that final sequence date of April 22nd. And so you can see that reported incidence right here as this horizontal line. So you can see that it's kind of most consistent with these very low pH values, meaning an incredible amount of kind of transmission heterogeneity or super spreading. And that if we assume lower levels of transmission heterogeneity, we wouldn't actually see the incidence that was, or predict the incidence that was actually reported. Okay, so we do see that there's kind of this indication of SARS-CoV-2 super spreading in Israel, and this has also been found kind of with more kind of epidemiological studies of viral spread in numerous different places. Okay, so getting to the second part of the talk, which is on tree-free phylodynamic inference. So this is very similar to that slide that Samuel had about kind of challenges in phylodynamic inference. So specifically, I'd want to focus here on the limitations of tree-based phylodynamic inference. For an emerging viral pathogen like SARS-CoV-2, right, and relatively low substitution rates and so forth, there can actually be a huge amount of phylogenetic uncertainty, right? And of course, you need to integrate over that phylogenetic uncertainty to get these epidemiological parameters that we're actually interested in, right? And you also need to estimate a lot of tree parameters as well, right? This all makes it very computationally very intensive, and scaling up to a much larger number of sequences becomes very difficult. Then with respect to birth-death models, they have somewhat more difficulty incorporating nonlinear disease dynamics, for example, kind of in SEIR type models. And then in terms of the tree-based inference approaches, they've also, I haven't seen kind of a very straightforward way to consider kind of multiple independently introduced clades at the same time. So either research that has been published has focused on a single clade, right? Or has tried to kind of incorporate multiple clades, for example, by actually modeling an outside reservoir class, like I mentioned. So these are kind of the limitations, I think, of tree-based phylodynamic inference for SARS-CoV-2 and for other kind of emerging viral pathogens. And so the question is, can we consider skirting tree-based phylodynamic inference for some of these kind of low diversity emerging pathogens, but still make use of viral sequences using a tree-free inference approach? And so the way we kind of started thinking about it was actually with this population genetic statistic, Tajima-D, that I think everyone has probably heard about already, at some point in their career. So just as a refresher, Tajima-D is the difference between two measures of genetic diversity, the pi, so the mean number of pairwise differences, and the number of segregating sites. It's oftentimes used for identifying selection, but it's very well known that under neutrality, population dynamics also impact the dynamics of Tajima-D, right? So expanding populations have negative values of Tajima-D, and populations that are contracting in size have positive values of Tajima-D. And so when we started thinking about this, we actually found this paper that was published in Epidemics back in 2017, which was actually trying to use kind of Tajima-D to infer, basically do phylodynamic inference. And we read this paper and we thought it was really interesting. Here's just kind of a summary of some of what they found. So what they did was they used ABC, which we all know about from Samuel's talk. So they used ABC to estimate epidemiological model parameters. And then they compared their results to the birth-death skyline serial model of Tanya Statla's. And what they found here on simulated data, where the true R naught was here at around 1.5. This is kind of their posterior and 95% credible intervals, and they found they were able to kind of recover the true R naught effectively. So that's on simulated data. And then this is an example kind of on pandemic H1N1, and they estimated an R naught of 1.55, using their Tajima-D approach, and the birth-death skyline model actually, which used these same data, had an estimate of 1.63. Anyhow, so they thought it was kind of a good alternative to the birth-death model. So we were interested in this, but I'm, even though I've kind of used ABC, I'm more partial towards likelihood-based phylodynamic inference. And so, we'd been thinking, well, maybe we can actually swap out the ABC portion kind of the Kim-et-al approach, using Tajima-D, and actually incorporate it within a particle MCMC inference framework. I don't know how many of you have heard about particle MCMC. Basically, it was developed by Andro and colleagues back in 2010. It incorporates particle filtering or sequential Monte Carlo into MCMC. I think of it as kind of the Bayesian equivalent to MIF, if some of you might know MIF, of Ed Ionides' and Aaron King's POMP package. So it's basically, if you're interested in reading about particle MCMC, this is kind of the definitive source. The underlying modeling framework for under this particle MCMC is a space modeling framework. So you have a process model and an observation model. You therefore also kind of estimate parameters, and you can reconstruct unobserved variables of that process model. And it's starting to be more heavily used by infectious disease modelers for interfacing epidemiological models with time series data, primarily of cases. Here's a nice kind of article that kind of talks about particle MCMC in terms of epidemiological inference. And kind of incorporating with sequence data allows for a very straightforward extension to combine, epidemiological case data, as is detailed here, with actually genetic data, right, sequence data. Just some preliminary results here. So this is from an underlying SEIR model, so susceptible, exposed, infectious, and recovered. Here, what we have is we're simulating, sequence evolution under an infinite sites model, and then kind of every 4 days, we're sampling 30 sequences from this underlying population. And we're assuming that sampling is occurring as individuals recover from being infectious. So we can get the simulated Tajima-SD time series, as you can see here. And so using particle MCMC, what you can see here as kind of the underlying simulated data here in red. Hopefully you can see that. Here we have the fraction of the population that's susceptible. Here we have the number exposed. Here we have the number infected. And here we have that Tajima-SD, which is reproduced right here. And so what we're trying to estimate is both the start of the simulated epidemic with a single first case, so it would be the clade introduction, right? And trying to estimate R naught. And so what you can see here are these posterior distributions for T0 and for R naught with the true values shown here in red. We have a prior on T0 and a formative prior, but we have a uniform prior on R naught. And so we can kind of recover, these, this epidemiological parameter of interest pretty well. Here, what you can see with the black lines are just kind of those reconstructed state variables from the process model. And so you can see that we can kind of, see what's happening to those dynamics as well. And you can see that they kind of hug kind of the true simulated values, true simulated data. Okay, so what we've been thinking about much more recently is, so it seems like Tajima-SD is somewhat informative, and we really want to kind of push the boundaries of kind of what's possible there, in terms of using it for tree-free phylodynamic inference. But we've also been thinking about, well, can we actually, instead of Tajima-SD, could we just use the site frequency spectrum, right? And, many of you are probably aware of kind of, the site frequency spectrum being used for demographic inference, not related to viruses, but for things like estimating human population size changes over time from a set of samples that are taken at a single time point. Here's just some of those papers. I noticed that some of these folks are actually here at this meeting. So we've been thinking about kind of incorporating the site frequency spectrum, which we can simulate right into this particle MCMC framework to estimate epidemiological parameters. The one issue with the site frequency spectrum is just in terms of being able to calculate the likelihood. It becomes more difficult than for just doing it with Tajima-SD. So we're still kind of working through that a little bit. And so just to summarize, kind of using this tree-free inference approaches, the benefits of that, is that it allows for very large sequence data sets to be considered because you don't have to actually estimate a tree or integrate over that phylogenetic uncertainty. And some initial work we've done in terms of considering multiple clades. So those can be easily considered because you then have multiple Tajima-SD time series or multiple SFS time series in one epidemiological model. And you're estimating parameter, same set of parameters, one epidemiological model, but multiple time series to interface it with. Incorporation with time series data on cases, for example, would be straightforward. And there's no really restrictions on the forward model considered, so you can use kind of an arbitrarily complex compartmental type model or something else. And then, we were thinking also like, well, it might be useful for something like SARS-CoV-2, but once SARS-CoV-2 gets enough genetic diversity, do we want to go back to kind of coalescent-based approaches, for example? But it seems like there's always kind of these questions popping up where there's something that has kind of, low degrees of genetic diversity. For example, like a new lineage coming up, like B117, initially that has very low levels of genetic diversity, right? And so, some of these methods actually being useful in those circumstances. And then, when you're thinking about kind of the site frequency spectrum, could some of these approaches also be applicable to phylodynamic inference for viral pathogens that actually undergoing extensive recombination, so where you don't actually have to consider, or you don't even want to consider full linkage, complete linkage. Okay, and with that, I'll finish up, and again, just thank and acknowledge my collaborators on the work that I presented, and I can take any questions.
Okay, so thank you very much, Katia, for this great talk. So indeed, we welcome questions. You are free to ask questions both in the chat or to speak up. If someone wants to speak up now, you are most welcome.
Hi, Katia. Very interesting work. This is
Hi, Michael. Good to see you.
I wonder, for this use of Tajima's D, did you consider the effects of subsampling? This is probably an issue in many populations, but in particular, of course, it has been highlighted in this SARS-CoV-2 thing, where it even varies between different subtrees. So it would appear to me that maybe the number of variants and the diversity react differently to subsampling. Is that something you look at?
Absolutely, yeah. So that and I brushed over that completely. So yeah, so we actually, by looking at kind of what happens to Tajima's D kind of over time, we also looked at different sampling schemes, and it turns out the value of Tajima's D is sensitive to the number of samples you take. Okay, so basically what we're doing in the particle MCMC is that we are effectively kind of matching the number of samples, right, from a given time window, and so that kind of gets around that. But yeah, but we don't. Yeah, so that's a very important part of it, because if we were to sample, large number of kind of sequences in our simulation, right, and basically figure out what the the probabilities of seeing the observed Tajima's D given kind of what's simulated, you need to kind of control for the number of samples for sure.
Thanks.
Thank you. Any other question? Again, feel free to speak up. So actually, I have a probably naive question, but I was interested by the fact that your Tajima D decreases in time in your simulated data. So what's the main ingredient for this?
Hopefully, hold on, let me see if I can actually pull up so it has to do with, whoops, I don't know if I'm gonna actually be able to find it right now, but it has to do with the dynamics of the number of segregating sites and the dynamics of pi, right? Since it's the difference there. And so since it's kind of pi minus, the number of segregating sites divided by this, this other parameter. So basically the number of segregating sites actually kind of is higher, right, than pi. And therefore it kind of goes negative when you have an expanding population.
I see, okay, yeah.
Yeah.
Cool, thanks. All right, so any other question in the chat? No. So, I mean, if not, we are going to continue the discussion in our Gather virtual space.